happy to have her as the speaker today. So Jayne, now is the time to start this webinar.
Thank you very much, Gregory. Good morning, everyone. It's time to go ahead and get started. And today we're going to talk about optimizing product development and production of gluten-free baked goods. So just a little bit about the instruments we're going to talk about today for gluten-free applications. We are specifically going to talk about the Rio F4 and the MixoLab 2. So the Rio F4 is designed to predict behavior during the fermentation process. So the way that the Rio fermentometer works is that there's a weight on top of a mass of dough. And during the fermentation process, you can actually measure the height increase of the dough during fermentation. In addition to measuring the height, or the increase in volume, we actually measure the production of carbon dioxide gas, and we can measure how much is retained by the dough over the course of fermentation and how much is lost over time. And it gets plotted onto a chart like this. The MixoLab 2 is more about evaluating mixing and baking behaviors of the raw materials that you're using for your formulation. So the MixoLab 2 is a recording mixer, and you have the ability to apply a heating and a cooling cycle to a mass of dough so that you can see the entire mixing and heating and cooling behavior. Now typically when we're talking about a wheat flour and water dough, we're measuring 6 dough quality criteria. We're measuring the water absorption of the wheat flour. We're measuring the effects of mixing, so examples are development and stability. We're measuring the gluten strength, the maximum viscosity during starch gelatinization, amylase activity, and the breakdown of the starch, and then starch retrogradation during the cooling phase. And this is an example of a standard MixoLab curve with a wheat flour and water dough. The curve looks largely similar for gluten-free products, but there are some notable differences. In general, the first part of the curve when we're talking about wheat flour can be ascribed to the gluten properties. In the case of gluten-free dough, that would be your structuring agents. And then the second portion of the curve is typically dependent on the starch properties, and this is the same for gluten-free formulations as well. So typically, water absorption is measured at the very front end of the curve, as you can see right here. And then the first 8 minutes measure the mixing properties of the material. The first 8 minutes is a measurement of the mixing properties of the material at 30 degrees Celsius. At that point, we apply some gentle heating, and you start to see, in this case, when we're talking about wheat flour, the properties of the gluten proteins and a weakening or a softening of the dough. And then as the proteins are denatured, you start to see the properties of the starch through gelatinization, breakdown, some amylase activity, and then during the cooling cycle, you'll see the setback or the retrogradation potential of the starch properties. So how can these instruments be used to help with gluten-free applications? Well, baking trials and analytical instrumentation go hand in hand. Now, baking trials are always the gold standard, but they're expensive. They require expert test bakers. Typically, these tests take a lot of time, and they can be very dependent on the operator performing the test. So the use of these instruments, these solutions, can help reduce the number of baking trials. And this allows you to focus more on challenging formulations and not trying to test and discriminate between good versus bad samples. They're also there to help by providing an objective and accurate result, meaning it's more operator independent. Obviously, you won't always have the same test baker there every day, Sometimes your test baker is sick or retires, and so this is a way to get away from operator-dependent results. So the gluten-free market is still a very strong and growing market. It is still expected to grow by about 10% per year from 2017 to 2025. And the reason is that people are eating gluten-free as part of an overall healthy lifestyle, not just due to dietary restrictions. And so by the year 2020, the gluten-free and overall free-from food retail sales just in the U.S. are predicted to hit about $24 billion. So this is going to be a huge market segment for the foreseeable future. Now let's talk about some of the key ingredients that go into gluten-free products. So first, we're working with new raw materials. Most of the time, our knowledge has been based on wheat flour, but when moving to gluten-free, we definitely have to get rid of the wheat flour and look at things like rice and corn and buckwheat. All of the non-gluten containing cereals and pseudo-cereals form the base of these formulations instead of wheat flour. Second of all, you have to have some kind of structuring agent in the absence of gluten. Now, this could be hydrocolloids like HPMC and carrageenan, or it could be an external protein source, for example, soybean proteins, egg proteins. And what these do, besides providing structure, is they increase the absorption of water, and then they provide volume to the product, especially when we're talking about bread products. Now, targeting the optimum water absorption level is critical for gluten-free product quality. So gluten-free doughs are much more hydrated than a conventional dough. So most of the times you're not working with a dough in the traditional sense of a bread dough. You're working with something that's more of a batter type of system. It's very highly hydrated. And then all gluten-free products, of course, aim to reach the quality of the corresponding conventional wheat flour containing product or gluten containing product. So you're still trying to achieve the same end result. You're just constructing the formulation differently to get there. So let's talk about the use of Mixolab 2 in gluten-free products formulation. So there's a large number of things that you can do with the Mixolab. First of all, just as with wheat flour, you can use it to select the best alternative raw materials and then control the consistency and regularity of the incoming raw material quality. You can also adapt the formula depending on the varying quality of the raw materials, for example, adjusting the water absorption level. And then you can also adjust mixing and baking properties as well. It can help you target the right ingredients to replace gluten functionalities depending on your process and your product. You can analyze the effect of different enzymes, especially those that are designed to strengthen the dough or the batter. You can analyze the rheological properties, and you can check the consistency and regularity of gluten-free mixes. Those are just a few of the applications. So let's look at the analysis of raw materials. So if we're getting away from wheat flour, we're looking at different raw materials, and they all have different rheological behaviors. Rice performs very differently from corn or maize, and that performs differently from buckwheat. And so you'll see in these graphs here, we have examples of buckwheat, both refined and whole grain. We have corn or maize, down in the lower left-hand corner, and then potato starch in the lower right-hand corner. All of these materials can be analyzed with the Mixolab. You don't have to modify the instrument, and you don't need to purchase an additional accessory. The instrument in its current form can be used right away to analyze non-gluten containing raw materials. So what you're really doing is getting information about the rheological behavior. And using that, you can predict some of the properties of the final products. And so If we look here in the upper left-hand corner, that's a Mixolab profile of wheat flour. So you can see in the lower right-hand corner, the Mixolab profile of rice flour is relatively similar to the Mixolab profile of wheat flour. So if you were looking to mimic the rheological behavior of wheat flour, that might be one way to go. If you look in the lower left-hand corner at the Mixolab profile of amaranth flour, looking at the rheological properties there, you probably don't want to mix this long in your process, for example. So it's using this curve and understanding the rheological information it's telling you to alter your process and choose raw materials, for example. So looking at the determination of optimum water absorption. So as I mentioned earlier, most gluten-free doughs aren't actually doughs so much as batters. They're very highly hydrated. However, when you look at different raw materials, the same hydration level doesn't necessarily translate to the same rheological properties. So what we're looking at here in this figure is we're looking at 65% water absorption, but we're looking at 65% water absorption in buckwheat, corn, and rice flours. So you can see the rheological properties change quite substantially at 65% water absorption, depending on the raw material that you're using. Now, if we look at just rice flour and we look at the effect of increasing water absorption, we can see that the mixing portion of the curve changes quite substantially. At 55% hydration, we have very stiff mixing, high consistency dough. At 90% hydration, we definitely have a very thin batter system that doesn't really show much of a mixing curve. And of course, this also has implications for starch gelatinization and retrogradation. So the water level greatly impacts the mixing behavior, as well as the gelatinization, gel stability, and setback. And so you can use the Mixolab to figure out what range of water absorption you need to target depending on your process and your product. Now, if we look at buckwheat dough, for example, there's a little bit more protein to add a little bit more structure there, so you can get a little bit of a mixing curve. And what I would point out here is that increasing water absorption does not result in a strictly linear relationship for all parameters. So it's important that when you're using a non-gluten containing flour that you study the impact of hydration across a wide range of hydration so that you can see which parameters behave more linearly, for example, C3 point, and which behave a little bit more logarithmic or curvilinear, for example, the C2 and the C1 points. And so using that, you can start to dial in on the right hydration level for the raw material that you're using, and you understand what happens to the mix as you add or remove water, and you can adjust your processing appropriately. So let's talk a little bit about structuring agents, hydrocolloids, and their effect on dough rheology. So when you remove the gluten from the product formulation, you obviously have to add something to serve as a structuring agent, and hydrocolloids are frequently called on in gluten-free formulations. Now, there are a lot of different gums and hydrocolloids available on the market, and depending on where you need the structuring effect determines which hydrocolloid or structuring agent that you choose. So, for example, HPMC and carrageenan increase dough consistency, as you can see from the graph on the left, and they give rise to higher stability in the dough. Now, xanthan, as most of you, if anyone has worked with xanthan gum before, it requires heating before you see a significant structuring effect. So you can definitely see with xanthan that as you get into the heating phase, you start to see the effect of adding xanthan gum. So depending on where you need the additional structure, you would target different hydrocolloids and structuring agents, and you can confirm that they are adding structure at the appropriate place in the process with the Mixolab. Now, what we're looking at here is we're specifically looking at HPMC added to rice dough in increasing quantities. It is a good structuring agent, and as you can see, as you increase the amount of HPMC, you're increasing the consistency of the rice dough during the mixing phase. And there is a two-step, you can see the two-step gelation process with HPMC. The first phase occurs at 30 degrees Celsius, which is the temperature of the mixing phase, and you can definitely see the increase in consistency there. But you can also see the increase in consistency as you start heating and you hit 75 degrees Celsius. So you see that additional structuring during heating contributed by the HPMC. And so by understanding where those shifts occur and to what extent, depending on the amount of HPMC is there, you can start to figure out and dial in on how much of a structuring agent you need to add. So let's take a quick look at enzymes and their influence on rheological properties. So we're looking at a combination of factors here. We're looking at soybean protein as a structuring agent, and then transglutaminase enzyme as a strengthening enzyme. So by adding the soybean protein, we're adding that structuring agent and improving the dough properties, especially in the mixing stage. But the addition of transglutaminase, while it doesn't have an impact in the mixing phase of the curve, you can see that during heating, the effect of transglutaminase becomes quite apparent, and it can enhance consistency to the extent that it matches the consistency of soybean protein at a 13% level. So if you're trying to reduce the amount of a structuring agent, especially a protein-based structuring agent that you're adding to save on formulation costs, by adding a little bit of an enzyme, you can see where in the process that enzyme will have an effect and to what extent. So what we're looking at here is the combined effect of transglutaminase and glucose oxidase on a buckwheat dough. And so, again, we're using these enzymes to strengthen the dough. And again, you don't see much of an effect during the mixing phase of the curve, but during heating, you certainly see an increase in the consistency depending on the combination of enzymes that you're using. So you can also use the Mixolab to look at full formulas, either within your own process to gauge consistency, or you can do a competitor analysis of some of your competitors' products, for example. So if we're talking about your own internal usage, you're probably looking to control consistency of your gluten-free mixes. The strategy is to design a specific gluten-free mix protocol that's appropriate for your product and your process. And so by having that, you basically have a QC tool to ensure the regularity and conformance of your gluten-free mixes. So in this case, this is an example protocol of, for a gluten-free analysis. And we were looking at a wide range of gluten-free mixes. Most of them were specific to bread, but we did evaluate other products as well. What we'll look at today are the breads. And in this case, what we would be looking at is more of a competitor analysis. So looking at 4 different gluten-free bread mixes, and of course they had a wide range of formulation strategies that they pursued, as you can see here by the ingredient list. And so what the profile shows is that there's a very wide range of rheological behaviors for each of the mixes, and that goes back to the formulation strategies that were used. For example, bread 3 showed the highest maximum torque at 30 degrees Celsius, and the highest, while breads 2 and 4 exhibited much lower maximum torques during the mixing stage. However, by the time you get to the end of the cooling cycle, most of the bread mixes showed very close final torques, so the amount of retrogradation at the end was very similar. You can also use it to, as we discussed, analyze incoming raw materials. And so in this case, what we're going to be looking at is a set of rice flours that were evaluated for gluten-free cookie quality. So the objectives of this small study were to evaluate the impact of rice origin on cookie quality, look at the rheological properties with the Mixolab, and identify correlations between the characteristics of the finished cookies and the Mixolab results. So we had a range of 6 commercial rice flours, and we used the same gluten-free protocol for evaluating all of these rice flours. In this case, we were hydrating to 70%. And then we also baked cookies. So it was a laminated dough sugar crust pastry product at a constant hydration. And of course, this follows pretty typical cookie protocol in terms of size of the cookie and baking. So looking at the results, you can see very clearly that each of the rice flours had an impact on the final product quality. We see differences in color and appearance, and we also saw differences in the thickness or the height of the cookie, the dimensions, and the weight of the cookie as well. And so looking at this, you can very clearly see that not all rice flours give you the same final product. And looking at the Mixolab profiles, you can definitely see there's a distribution of rheological properties across all of the different rices, or rice flours, I should say. For example, rice flours 3 and 4 were relatively close in consistency at the mix stage. Rice flour number 6 had the lowest cold consistency, but it did gel up and create a very similar consistency during the heating phase. So you can see the behavior here is quite different, and this translates into differences in the cookies. And so looking at the correlations, cookie weight is highly correlated with the C5 point, which is basically the end of it's the starch retrogradation, it's the end of the test cycle. And then also mixing stability. And so cookie weight is going to be related to how much water that the product retains at the end of baking. So it makes sense that the final viscosity at the end and the stability would be related to that. Cookie thickness was correlated with the C4 point as well as development time. So the C4 point is the breakdown in the curve. It's the breakdown during the heating cycle, and the development time is related to protein or structuring agent strength. And so it makes sense that cookie thickness would be correlated to how much structure both the starch and the structuring agent can provide. So as you saw, we saw differences in color and physical properties. The Mixolab showed very different rheological properties from the different rice flours, and the characteristics of the finished products correlated very well with the Mixolab data. So you could use these results, for example, to help choose raw materials, ensure the consistency and regularity of those raw materials. Maybe you use it to improve or modify existing formulas, or you use it to develop new products adapted to a specific process or adapted to needs of consumers. So let's take a quick look at the Rio F4 and some of its gluten-free applications. So just like the Mixolab, the Rio F4 can be used for the selection of ingredients, especially structuring agents, in terms of giving the best volume during fermentation. They can help to ensure the quality and the quantity of the ingredient to use, and of course, check for consistency of that ingredient performance. You can also use the Rio F4 to determine optimum process properties, for example, mixing time, mixing speed, particle size, in terms of achieving the best volume in the final product. You can measure the impact of water absorption or water addition levels on the proofing properties, and evaluate the impact of other ingredients and just proofing performance in general of gluten-free mixes. So in terms of optimizing the baking performance, we can look at things like particle size, as I mentioned. So in this case, what we're looking at is we're looking at the effect of flour particle size on the proofing performance of a yellow semolina flour as well as a yellow maize flour. And as you can see, in both cases, as you decrease the particle size, the proofing performance decreases significantly. So you can look at the impact of milling the particle size on proofing performance. In terms of optimizing gluten-free bread volume, what we're going to be looking at is we're going to be looking at a rice flour-based formula mixed in a KitchenAid. It's more of a batter type system, and it's going to be measured. The proofing performance is going to be measured in the Rio F4 at 30 degrees Celsius. And because this is a gluten-free formulation, I wouldn't recommend using weights since, as I mentioned, it's more of a batter system and not a very firm bread-like dough. So in this case, one of the things that we looked at was we looked at the influence of mixing time on proofing performance. And as you can see, as you increase mixing time, you significantly reduce proofing performance. So in this case, this would be a process optimization, reducing mix time to somewhere around 2 minutes in order to achieve better volume in the final bread product. So to start to wrap things up, the Mixolab and the Rio F4 can help selecting the best raw materials and controlling their consistency, selecting the right structuring agents to replace gluten in the formula, controlling the consistency of gluten-free mixes, determining the optimum water absorption level, finding the recipe with the best expected shelf life, and adapting the formula depending on the quality of the raw material. These are just a few things they can help with. There are numerous companies that already use CHOPIN Technlogies solutions. Some of those would include Ingredion, Delavo Food Partners, IATA, and University College Cork. So I'm sure they would be very willing to chat about how they've been using the instruments. And just to put a few more conclusions out there, what you get from these solutions is the control of raw materials, help in selecting and dosing the right ingredients and additives. You can anticipate and adapt to changes in processing properties. You can reduce the number of bake tests, and you can speed up the new product development process. So if you want more information or are interested in a trial, you are welcome to contact Gregory Vericel at the email address indicated in the slide here, so gvericel@chopin.fr. And there are a whole host of solutions that CHOPIN Technlogies offers for the baking industry, not just gluten-free. So frozen dough, whole grain, salt reduction, sugar reduction, clean label. There are all kinds of solutions that you can pursue with the assistance of CHOPIN. All right, I'll open the floor to any questions.
Okay, thank you, Jayne. This is Bob Schumann again. I would remind everybody that there is a Q&A panel in your WebEx screen there. So I've got a couple of questions, but please type in any questions that any of our attendees have. And with that, I will go to our first one. And the first one is, how do you determine optimum water absorption? So I'll put that out for Jayne or Gregory if this is appropriate. So how do you determine optimum water absorption?
So one of the things that you need to do is you need to determine the optimum consistency for your process. So typically you're going to be working with a process limitation in terms of batter consistency. So once you know the consistency that you can work with on your processing line, the next step would be to measure that in the Mix-a-Lab and then target that, that consistency in terms of water absorption with the formulation that you're using.
Okay. And another one on the Mix-a-Lab also, what is the recommended and minimum dough weights for the Mix-a-Lab? Does it always have to be 75 grams?
Well, typically when you're working with the wheat flour dough, the recommendation is to start with 75, or to target 75 grams. I don't know if you noticed the protocols, the example protocols that were denoted in the slides, but with gluten-free, we were using 90 grams. There have been instances where we've increased that to 100 grams of dough, so you certainly can increase the dough weight to improve the consistency of the results that you're getting from the Mix-a-Lab. I wouldn't recommend going much lower, simply because you have to maintain a minimum fill level in the mixer to guarantee consistency of your results. So I would say, especially with gluten-free, you don't really want to go much lower than 75.
Okay. Another one on the Mix-a-Lab. How would you predict shelf life of gluten-free products?
So in the Mix-a-Lab curve, the last portion of the curve, you see the retrogradation during the cooling cycle. That's typically the most correlated to texture results. So what you would want to do is measure texture of your product over time, typically a firmness, and correlate that with the retrogradation portion of the curve. And you may also find some correlations with the C3 and C4 points in the curve as well, since those are also starch properties.
Okay. How long does it take to create a new protocol or a new method on the Mix-a-Lab?
So it really doesn't take that much time. It's a matter of maybe 5 minutes or so. The key is knowing which parameter of the protocol that you want to change, whether you want to change speed or you want to change the temperature profile, or even if you're changing both. So changing the protocol itself doesn't take much time. It's knowing what parameters that you want to target and how you want to target them.
Okay. I've got one final one. And again, if anybody has any questions or need to elaborate on these answers, please put them in the Q&A section quickly. The last one is about the experiment that you described with the cookies with the rice flour. It's asking, what is the recipe? So could you describe the formulation or the recipe about that rice flour cookie experiment?
Yeah, Jayne, if you don't mind, I can answer that one.
Certainly.
That's a study we did with the French Institute of Baking. So they developed a specific protocol for cookie baking tests. So it's pretty standard sugar crust pastry, and the way they do that, they do 3 different mixing steps. During the first step, they add butter, sugar, and salt. So it's about 15% butter, 15% sugar, and just a little bit of salt, 0.2, 0.3%. In the second mixing stage, they add eggs, so it's about 15% in weight. And the last mixing step, they add flour, about 50%, and yeast. And after that, they follow the protocol that Jayne explained regarding the lamination, the cutting, and all the pretty standard steps for cookie baking tests.
Okay. Okay, I think I don't see any further questions. If any of the attendees have any further questions, or they want to pass along questions to Jayne or Gregory, you can contact me. You should have my email. It's B as in boy Schumann at unityscientific.com. I'll be happy to pass them along to Jayne and Gregory. And with that, I'd like to thank everybody. Thank you, Jayne and Gregory, for presenting this, and love to thank all of the attendees for joining us. So thanks, everyone. Have a good morning or afternoon. Thank you. Bye-bye.
Thank you, Bob. Thank you very much.